Have you ever wondered how people can look different? Even siblings have distinct differences between them, unless of course you're an identical twin. Why is that? How can people with the same biological parents look different from one another? This is because of a term called genetic variability. In today's video, we will discover two sources of genetic variability: crossing over and independent assortment. Both crossing over and independent assortment are produced by meiosis. Hi, I'm Faith from Full Potential Learning Academy, and today we are going to discuss some important facts about meiosis. But before we start, let's cover the cell cycle briefly. The cell cycle is composed of interphase and mitosis. The cell cycle is responsible for maintaining an appropriate amount of cells within our bodies by dividing one cell into two daughter cells, and this happens all around our bodies. If you'd like to learn more information about the cell cycle, you can click this screen here. In mitosis, two identical daughter cells are produced from one parent cell. Both the parent cell and daughter cells are diploid, meaning that they have two non-identical sets of chromosomes, one from each parent. Remember, this only occurs in somatic cells, or non-sex cells. What are somatic cells? Somatic cells include skin cells, liver cells, heart cells, fat cells, and more. In meiosis, 4 daughter cells are produced, unlike the 2 in mitosis. However, the daughter cells are not identical to the parent and only contain one set of chromosomes and are termed haploid. In this picture, I colored the cells different colors to show that the daughter cells are different from the parent cell. However, the real difference is determined by processes called crossing over and independent assortment. We'll talk more about that later on. Meiosis, like mitosis, is preceded by interphase. Interphase contains 3 stages: G1, S, and G2. Just like in mitosis, G1 and G2 are the phases in which the cell grows and prepares for mitosis. In S phase, or synthesis, the cell duplicates its DNA content, which occurs within the cell. Since there is a reduction of chromosome numbers from diploid to haploid, meiosis includes one round of chromosome duplication, which occurs in interphase, and 2 rounds of division. The first round of division is called Meiosis I, and the second round of division is called Meiosis II. Looking at this slide, do you see anything familiar? The events that occur in meiosis are analogous to the events in mitosis, so the same stage names are given in meiosis as mitosis. Meiosis I. In prophase I, crossing over occurs, the cell and its contents condense or disintegrate, the nuclear envelope, or the barrier surrounding the nucleus, breaks down, and centrosomes begin to move to opposite poles. The first event of genetic variability is crossing over. Crossing over occurs only in prophase I. The pairing of homologous chromosomes is called a synapsis. In synapsis, the genes on the chromatids of homologous chromosomes are aligned precisely on top of each other, as you can see in the picture on the left. You can see a red homologous chromosome on top of a blue homologous chromosome. During this time, the homologous chromosomes create a synaptonemal complex full of proteins. The synaptonemal complex supports the exchange of chromosomal segments, or DNA, between non-sister homologous chromosomes. Located along the synaptonemal complex are large protein groups called recombination nodules. These groups mark the points of later chiasmata, or singular chiasma. A chiasma is a specific location of contact between homologous chromosomes, where DNA information is exchanged. The number of chiasmata varies according to the species, but there must be at least 1 chiasma per chromosome for proper separation of homologous chromosomes. However, there can be as many as 25 chiasmata. A single crossover event between homologous non-sister chromatids leads to an exchange, or recombination, of equal DNA between a maternal chromosome and a paternal chromosome. So the chromatid from one parent and the chromatid from another parent exchange equal DNA. This exchange will create two chromatids called recombinant chromatids. The chromatids that didn't exchange DNA are called unrecombined chromatids. Following crossover, the synaptonemal complex breaks down, and at the end of prophase 1, the pairs are held together only at the chiasmata and are called a tetrad because the 4 sister chromatids of each pair of homologous chromosomes are now visible. In prometaphase 1 and metaphase 1, the homologous chromosomes are finishing their condensation. Kinetochores appear on the centromeres, and mitotic spindles microtubules attach to them. The stacked homologous chromosomes align randomly at the metaphase plate, in a process called random or independent assortment, which is a second source of genetic variability. Remember that in meiosis I, the homologous chromosomes, as you can see in this picture, are held together only at the chiasmata. In anaphase I, the stacked homologous chromosomes are pulled to opposite poles. In telophase I and cytokinesis I, chromosomes arrive at opposite sides of the poles and begin to decondense. The nuclear envelope reforms and the mitotic spindle breaks down, and the cell splits into 2 haploid daughter cells. So some of these events depend on the species, so some species may not decondense their chromosomes, or some of their nuclear envelopes will not reform. It all depends on the species. Meiosis II. In prophase 2, the cell and its contents condense or disintegrate if they did in telophase 1, the nuclear envelope breaks down, and centrosomes begin to move to opposite poles. In prometaphase 2, the sister chromatids continue condensing and are getting ready to align at the metaphase plate. The mitotic spindle develops and spindle microtubules lengthen outwards from the centromere. Each sister chromatid develops a kinetochore in their centromeric region. The kinetochores were attached with spindle microtubules. Remember that in meiosis II, sister chromatids are held together by a centromere, not by a chiasmata. In metaphase 2, sister chromatids align at the metaphase plate in a vertical line, not paired together like in meiosis I. In anaphase 2, sister chromatids are pulled to opposite poles. In telophase 2 and cytokinesis 2, chromosomes arrive at opposite poles and begin to decondense. The nuclear envelope reforms and the mitotic spindle breaks down, and the cell splits into 4 haploid daughter cells. Here are the major differences between meiosis and mitosis. Meiosis only occurs in reproductive structures, while mitosis only occurs in somatic cells. In meiosis, cell division occurs twice, while in mitosis, cell division occurs once. In meiosis, there's genetic variability, which occurs because of crossing over and independent assortment. But in mitosis, there's no genetic variability because the daughter cells are identical to the parent cells. In meiosis, 4 haploid daughter cells are produced. In mitosis, 2 diploid daughter cells are produced, identical to the parent cells. And finally, in meiosis, all daughter cells are non-identical, while in mitosis, all daughter cells are identical to the parent cell. Here's a summary of everything that we've learned. 1. Meiosis and mitosis both have 5 primary phases: prophase, prometaphase, metaphase, anaphase, and telophase/cytokinesis. 2. Unlike mitosis, in which these phases occur one time, in meiosis they occur twice, in meiosis I and meiosis II. 3. The overall action in meiosis I is to separate homologous chromosomes, while the overall action in meiosis II is to separate sister chromatids. And 4. There are 5 major differences between mitosis and meiosis. Here are some questions for you. 1. During what phase does crossing over occur? 2. Why do you think sex cells have to be haploid before fertilization? And 3. How can the process of meiosis generate genetic variation? I hope you enjoyed this video. Bye!